Microbiology plays a critical role in the biopharmaceutical manufacturing process. In this course, we will explore the relationship between microbiology and contamination, and the impact it has on the production of a safe and effective consumer product. Let's begin with an examination of contamination as it relates to pharmaceutical products and explore how microbiology relates to biological contamination. There are several types of contamination and risks associated with unchecked contamination. In many cases, contamination is biological, which means it involves the presence of unwanted microorganisms. We will discuss some of the microbiology around this type of contamination. Having a basic understanding of biological contamination leads to an understanding of what type of corrective action to take. First, let's define contamination and how it relates to a pharmaceutical product. Contamination is the unwanted introduction of an impurity. This impurity can be something physical, such as a particle of dust, or it can be more complex, such as the unwanted introduction of a biological organism. Take a second and read this definition. Contamination comes in many forms: physical, chemical, or biological. Physical contamination includes fiber material, particles, and chips from tableting tools. Chemical contamination includes vapor, gases, moisture, and molecules. And biological contamination includes fungus, bacteria, and viruses. During pharmaceutical manufacturing, great lengths are taken to prevent one product from contaminating a second product. This would be a form of chemical contamination that is often referred to as cross-contamination. Cross-contamination is possible when the unwanted matter is introduced or brought from one process to the next during manufacturing. So how can we unwantedly introduce contamination? People are a primary source of contamination. It can be transmitted by touch or brought into a process from our clothing. In some cases, the equipment itself can be a source of contamination, especially if it has not been cleaned properly. We have all heard of Legionnaire's disease. This was caused by bacterial contamination of the cooling towers used in commercial HVAC or heating, ventilation, and air conditioning systems. The bacterial contamination leads to the development of atypical pneumonia in people exposed to this organism. A mix-up is also a chemical form of contamination, but in this case, it is a human error where the wrong compound or the wrong strength of compound is used during production of a product. In pharmaceutical manufacturing, the risks associated become higher depending on the type of product and the type of contamination. Treatments involving living organisms, chemotherapeutic agents, or hormones are at high risk. Contamination becomes significant if it is a product that is expected to be put onto an open wound or injected. This could allow direct entry to a patient's bloodstream. Any contamination of a drug that is to be given over a long period of time is very significant, as the potential exposure to the contamination occurs over a greater length of time. Recently, contamination from COVID-19 has been a great concern. Controlling contamination, and therefore the spread of the disease, would have a positive effect on the healthcare system's ability to treat patients. The consequences of contamination with respect to the consumer can vary depending on the type of contamination and the route of delivery. For example, microbial contamination of something taken orally may just cause some nausea. But microbial contamination of a drug intended to be injected into the bloodstream of a patient can be fatal. Chemical contamination, regardless of how it happened, could seriously affect how well the product works or may even shorten its shelf life. Contamination can also affect the company making the product. The company loses the respect of the consumer and can be subjected to regulatory action. This slide shows examples of physical contamination. These can range from dust particles to metal flakes and fibers. As you can see on this slide, they include dust, dirt, rust, glass, metal particulates, fibers and hairs, and machine greases and oils. Chemical contamination is a little more complex. It can derive from the ingredients degrading prematurely, or from the process itself, like residual solvents, additives, or even cross-contamination with another product. Biological contamination can include bacteria, fungi, or viruses. The source of this type of contamination is usually from the environment or directly from personnel. Some of the common causes are shown on this slide, such as unhygienic and unsanitary practice, improper work attire, use of contaminated materials and equipment, open wounds or lesions in operators, and operators suffering from infectious disease. Personnel are the primary source of particulates in the clean room because we naturally shed particulates and microbes. For example, people shed skin cells, hair, foreign particulates, fungi, bacteria, skin oils, and viruses both into the aseptic processing area and into the product if proper aseptic practices are not followed. To prevent contamination of the product, good personal hygiene must be maintained. This includes taking a daily shower to reduce skin shedding, removing jewelry and makeup, and conducting proper hand washing prior to gowning. Sick employees or ones with compromised skin, such as a fresh wound or sunburn, should not enter the clean room due to increased risk of product contamination. The FDA has much to say about microbial contamination. The Code of Federal Regulations, CFR 21, has specific requirements especially for products that are supposed to be sterile. Unwanted contamination of these products can lead to serious health issues and potentially be fatal. Now that we have a better understanding of the general concepts of contamination, let's explore biological contamination in more detail. Bacteria, fungi, and viruses. Let's start with bacteria. They have been around almost as long as the Earth. There are possibly trillions of different species of bacteria here on this planet. They are very small, about 1/1,000,000th of a meter, and therefore cannot be seen with the naked eye. To help you visualize what a millionth is, imagine one piano key from all the keys of 11,363 pianos, or 1 second out of 11.5 days. Bacterial contamination can quickly get out of control because they reproduce by binary fission. This is another way of saying that one cell divides into 2 cells, and each of those divide to give 4 cells, and on and on. In some species of bacteria, this can happen every 20 to 30 minutes. From one single organism, you can end up with over 1 billion within 15 hours. This table shows just how astronomical the numbers can be if this biological contamination goes unchecked. In order for bacteria to grow, they need certain conditions to be met. These are just some of the factors that affect bacterial growth. Bacteria are found everywhere and can thrive in even the harshest of environments. If they find life on Mars, it is expected to be an ancient form of bacteria. These bacteria are known as extremophiles because they can exist where most things cannot. An example of these closer to home are the bacteria that thrive in and give color to the hot springs in Yellowstone Park. Here we see some of the sources of bacterial contamination. It is a major concern in the drugs that are supposed to be sterile or aseptic. Humans are the primary source of microbial contamination. This slide details just how many bacteria are in and on the average person. Take a few seconds to look at the sources and numbers. This represents the classification of bacteria called extremophiles. As you can see, some can tolerate extremes of temperature, salt concentration, and pH. They have found some bacteria surviving at the vents at the bottom of the ocean. Besides withstanding the heat associated with these thermal vents, they are subject to extreme pressure at the bottom of the ocean. Here is a picture of the colorful mats of bacteria that survive in the hot springs of Yellowstone. The water temperature here can be 160 degrees Fahrenheit. The mats represent colonies living together. The surface bacteria can use sunlight to make nutrients, which gets passed on to help fuel the organisms underneath. These deeper organisms decompose and give nutrients back to the surface dwellers. There are physical and chemical adaptations that allow for such extreme living. The physical characteristics of importance are the thickness and construction of bacterial cell walls. These differences can be visualized under a microscope using a special staining process called gram stain. There are gram-positive bacteria, which stain purple, and gram-negative bacteria, which stain pink. Gram-negative bacteria are medically important because remnants of their cell walls can cause fever and shock in patients. The gram-negative bacteria contain lipopolysaccharides, or LPS, in their cell wall, which are the fever-causing components. These are called endotoxins. There are strict regulations around their presence and concentration in products. Drug manufacturing processes must have ways to reduce and control endotoxin levels. This can be accomplished by testing, cleaning methodologies, and by depyrogenation using dry heat. Sources of endotoxin are anywhere that gram-negative bacteria can be found. Some of these sources include raw materials, water, excipients, non-active ingredients like salts and buffers, cell culture and fermentation additives, equipment, people, and anywhere gram-negative bacteria are found. Some bacteria can even ride out difficult conditions by forming spores. In this state, they can sit dormant and wait for the right conditions to return. Scientists have been known to revive bacterial spores that were over 2,000 years old. This slide shows the life cycle of bacteria that can form spores. One significant example of bacterial spores are the anthrax attacks. In this case, the anthrax spores were sent via mail packages. When the packages were opened, the spores were inhaled into the victim's lungs. There, they found the right conditions and sporulated, which means they went back into fully functioning bacteria. The living bacteria are notorious for forming a slimy coating and therefore quickly block the victim's lung function. To recap the importance of bacterial contamination, bacteria are worrisome because they can be found everywhere, multiply quickly, and can cause problems with the products or the patients who receive them. As we can see, there are many considerations which must be taken into account during the manufacturing process itself. Bacteria can grow in pharmaceutical water systems. Bacteria can survive continuous exposure to 80 degrees Celsius water and to steam systems. Bacterial endospores are extremely resilient. Some bacteria can withstand exposure to most disinfectants, moist heat, UV, desiccation, and irradiation. Gram-negative bacteria release endotoxins causing endotoxemia. Autoclaving does not destroy endotoxins. Sterile filtration does not remove endotoxins. Gram-negative bacteria requires depyrogenation at 250 degrees Celsius. Another pharmaceutically important biological contaminant are the fungi. Examples of good fungi include the blue in blue cheese and the many yeasts which help to make wines and beer. Mushrooms, such as the ones seen in the moist woods in the fall, are an example of visible fungi. Fungal contaminants can come from the ground or the air. In the lab, they can be grown on special gelatin-like agar plates that have nutrients in them. Most fungi will present themselves as fuzzy colonies of various shapes and colors. Yeast are also fungi, but they form nice rounded colonies when plated onto agar plates. Home examples of yeast use are for making breads, wine, and beer. Fungal contamination may be considered public enemy number 2 with respect to pharmaceutical manufacturing. Contamination by viruses are in a different league altogether. It is very controversial whether or not they are really the simplest form of life. They are basically cellular pirates. They cannot survive outside of a living cell, since they do not have the machinery themselves to reproduce. Instead, they contain the genetic material or blueprint to make copies of themselves, which is then surrounded by a protein coat for protection. Once inside a host cell, they direct the cell to stop whatever it was doing before and start making copies of the virus. This is why we get so sick in a viral infection. Viruses are very small, billionths of a meter in size. To put this in perspective, it takes about 1 billion marbles to equal the size of the Earth. Because of their size, and the fact that they do not directly metabolize anything, they are very hard to find in a pharmaceutical process. COVID-19 is caused by a respiratory virus that most likely started in a bat host. It then underwent mutations which allowed it to infect humans. It is highly infectious and can spread via fecal, oral, and respiratory routes. Viruses have very specific targets for attachment to a host cell. This virus attaches to one of the cells found deep in the lungs, where it destroys host cells and triggers catastrophic immune responses. By virus standards, the COVID virus, SARS-CoV-2, is a large-sized virus. In addition to the protective protein coat, the virus is surrounded by a lipid or fatty envelope. This envelope is easy to break down with normal detergents, which will hinder the virus attachment capabilities. The COVID virus is a corona-type virus, which means it has a crown of protein spikes radiating off its surface. This is an electron micrograph of the coronavirus, stained blue, in the lungs of the first U.S. patient. The virus spread quickly worldwide, affecting hundreds of countries. It is thought that each infected person had the capability of infecting 3 other people. This could happen approximately every 5 to 7.5 days, so we can see how rapidly this infection spread. It should be noted that as of May 8th, 1 month after this picture was captured, the pandemic stood at nearly 4 million cases worldwide, with over 250,000 global deaths. As you can see, controlling microorganisms is very important to the overall pharmaceutical manufacturing process in order to deliver a safe and effective drug product. The manufacturing and the release of drug products requires specific testing for contamination. Here are some of the testing required. Since biological contamination can spread so quickly, it's important to control their numbers during the entire manufacturing process. Even if there is a final sterilization procedure, an overwhelming number of microorganisms can hinder the ability to render the product sterile. The defining number of biological contamination at any one stage in the process is called bioburden. Sterility is the total absence of any microorganisms or their remnants. For products that are expected to be sterile, sterility testing is required by law. This is an example from the industry where product sterility testing is being performed. Because product release, patient safety, and ultimately product sales rely on the validity of this testing, it is being performed in a pre-sterilized half-suit designed to minimize risk of operator contamination, or a false positive result. As we can see from this slide, there are many considerations that must be taken into account when performing product sterility testing. The testing laboratory must be as good or better than the environment of the filling operations in order to minimize the risk of detecting false positives. The growth media used in sterility testing must demonstrate the ability to support growth. The entire batch must be represented when sampling for sterility testing. Now you know why microbiology and its relationship with contamination can have a significant impact on the biopharmaceutical manufacturing process. The topics and concepts covered in this course are intended to provide a foundation for your coursework, your success in this industry, and most importantly, the safety and efficacy of the products you produce.